Thank you, Rafael. First of all, muchísimas gracias, Monseñor Sánchez Orondo, por recibirnos aquí. Thank you, Eddie, for the invitation. It's a pleasure being here. So during this probably half an hour or 25 minutes, I'm going to show you some of the challenges in our field and some of the work we've been doing. So I'm going to talk about some sound perception and from the ear to the brain and then back again. So hearing loss is one of the main disabilities, sensory disabilities we're all challenged through. And millions of people worldwide suffer from hearing loss. 1 in 1,000 newborns have a hearing defect, and this increases with age, to the extent that by the year of 70 or older, 2/3 of the population will have some sort of hearing loss. And the problem with hearing loss is that it not only produces hearing loss, but it's accompanied by lots of other comorbidities. And people who have hearing loss and cannot hear, cannot communicate, they also sometimes get into depression. They cannot socialize. And with hearing loss and depressions comes cognitive impairment. And with the increasing of life expectancy, this is becoming a social and economic burden. So during this following slides, I'm going to try to show you some challenges that I think we are facing in the field, which still need lots of research. And these are, how do we prevent hearing loss? How do we restore hearing? And finally, which are the genes and molecules that are involved, both in hearing loss and in the normal functioning physiology of our hearing system. Now, up front, I'm going to tell you that restoring hearing loss is a huge challenge and something that we can't really fully accomplish. And so far, hearing loss can only be restored using hearing aids or cochlear implants. But all other let's say, biological means of restoring hearing loss are failing. And that is either stem cells or genetic engineering, gene delivery, do not work. And the fact that they do not work is because the cells that we use to hear, our sensory cells, do not regenerate. We have very few of them, and they are lost along our lifetime. And when we lose them, we still cannot regenerate them. So all attempts that are being done by using gene therapy or virus-induced gene therapy are not working. So the best thing that we can do is prevent hearing loss. So to prevent hearing loss, we need to understand what produces hearing loss. And hearing loss is produced mainly by two things. One is our genetics, the genetics of hearing loss, and the other is our ambient causes. And the main ambient cause that produces hearing loss is the exposure to loud sound. Loud sound is the main cause that leads to hearing loss. It's the main hazard that we are exposed in our daily lives in busy cities, where cosmopolitan cities that we all live. And how loud is loud? And you can see here that the exposure to hearing to loud sound not only depends upon the amount of sound or the sound level, that anything above 85 decibels is dangerous and can produce a permanent hearing loss that does not recover. But it not only depends upon the amount of sound, but also in the time exposure. So this does not show very much here, this pointer, but you can see here that 100 decibels, for example, or 100 and 110 decibels, thank you. This is much better. So you can see here that headphones, for example, from an iPod that our children are using, can be cranked up to 110 decibels. And 110 decibels, you can only be exposed for about 1 minute. Above this, you start getting some sort of permanent hearing loss. And this is accumulative. You start accumulating damage along your lifetime, and at one point, that does not recover. And hearing loss is not the only problem, because hearing loss comes with another problem which not everybody is acquainted of, which is tinnitus, or the perception of phantom sound. And a phantom sound is when we hear a noise in our head which is not produced by any external sound reproducer. The sound does not come from inside. We hear it in our brain, and this is very, very disabilitating, because it also comes with depression, anxiety, insomnia. So people that have tinnitus are sometimes really, really impaired to have a normal life. So as I told you at the beginning, we have the external exposure to sound, which is one of the main causes of hearing loss, but the other cause of hearing loss is our genetics. And hearing loss is 50% of hearing impairment is of genetic origin. As I told you at the beginning, 1 in 1,000 newborns have some sort of hearing impairment. 50% is because of genetic origin. It can be both syndromic and non-syndromic origin. 400 syndromes are known to be to include hearing loss, and from the non-syndromic hearing loss, we have all flavors from autosomic recessive, dominant, X-linked, etc. There's one gene that is the main cause of hearing loss, which is a connexin 26, but that only diagnoses, here it says 50%, it's less than 50%. Probably 20% of the people that have a genetic origin of hearing loss is due to mutations in connexin, but the 80% which are left without a mutation in connexin is produced by lots of other genes. Nowadays we know like 150 loci which have been linked to hearing loss, and we know about 100 genes which mutations produce hearing loss. And this paper came out last week in Nature Communications from the International Mouse Phenotyping Consortium, which concludes that there is an extensive unexplored genetic landscape for auditory dysfunction, and it is calculated that 400 different genes probably produced hearing loss. So when a patient comes to a genetist or to our lab and has a genetic origin of hearing loss and wants to know what gene they have a mutation in, it's very difficult, because you can see there are 400 different genes that some we know, some we don't know. So nowadays what it's being done at the clinics is whole exome sequencing, and then try to filter to find the cause of that hearing loss. Now, as the title of my talk was From the Ear to the Brain and Back, now I've posed what are the challenges. Now I'm going to talk about the molecules and the genes, some of them, not the 400, that we know that are important for not only hearing loss, but auditory physiology. Most of the genes that produce hearing impairment are expressed in the inner ear, in this organ, which is the cochlea. And the cochlea is the organ that has the sensory hair cells. This is a schematics of the organ of Corti, which is the sensory epithelium, responses for transducing sound stimuli into electrical signals that then are sent to the brain. This organ of Corti has different cells. The primary receptor cells are these pink ones here and these pink ones here. These are the inner hair cells, which are the phonoreceptors per se. The function is to transduce a mechanical stimuli at the apex of the cell into electrical signal that is then transported through this blue auditory nerve to the central nervous system. Now, the cochlea has inner hair cells, which are the primary receptor cells, but it has also 3 rows of these outer hair cells, which are not the primary receptor cells, but whose function I'm going to talk about a little bit in the second part of the talk, and this is where our work comes into play. Now, they're called hair cells because in their apex they have these different, what are called cilia, which are in the apex, and the anatomy of the cilia differs from species to species, from hair cell to hair cell. If we look at the organ of Corti of a mammal, we can see that the mammal has 1 row of this cilia here, which belong to the 1 row of inner hair cells, which are the phonoreceptors, and 3 rows of these outer hair cells, which have this V-shape like stereocilia. This is a normal epithelia, a scanning microscopy of a normal organ of Corti epithelia. Now, when hearing damage is produced and we are exposed to loud sound, 2 things happen. The stereocilia disarrange, and this can be reversible. But once we start losing hair cells, and you can see here the normal cochlea with the 3 rows of outer hair cells, and here there are hair cells that are missing. Once this occurs, this is irreversible because these hair cells do not regenerate. Now, how is this process of mechanical transduction produced? Sound stimuli comes in, it produces movement of the liquids in the inner ear. This produces movement of the tectorial membrane, movement of the cilia, and this produces the opening of channels which are at the apex of the hair cells, which are mechanically gated. This produces an influx of potassium and calcium, and depolarization of this hair cell, which leads to a receptor potential in this hair cell and release of neurotransmitter at the base of the hair cell and the activation of the auditory fiber. So how is this mechanotransduction produced? In the stereocilia, in the apex of the stereocilia, you can see that one stereocilia is connected to the next stereocilia by this fiber, it's known as a tip link. And when the cilia move, this tip link is thought to open a channel which is at the apex of the cilia, this mechanically gated channel. And since the stereocilia is based on the very rich potassium fluid, which is called the endolymph, potassium goes in and depolarization is produced. And when this, how does this mechanotransduction takes place? And this is amazing that in this post-genomic area, we still do not really know the structure of this mechanotransduction channel that is expressed at the apex of the hair cell. There are several candidates that have, there are now proposed trip channels, as Ramon was speaking about yesterday, were proposed initially to be the mechanoelectrical transduction channel in a very nice paper in Nature, and the same author a year afterwards published a Neuron paper saying that it was not the channel. So he got 2 papers, a Nature paper saying, I found the channel, and then a Neuron paper saying, no, it's not the channel. So still in this, it's very difficult to publish in Nature and Neuron, but he managed to do it, saying yes and then saying no. But still in this post-genomic area, we still don't know the structure of the mechanotransduction channel. So this is still a challenge in the biology of hearing. Now, once the channel is open, the cell depolarizes, and at the base of this cell, we know a lot of things. Glutamate is released, and this activates AMPA receptors, and then the auditory fiber depolarizes. So I've shown you how mechanotransduction is produced as the inner hair cell, but as I told you, the auditory epithelium has these other cells, which are the outer hair cells. And outer hair cells are not the primary receptor cells, but they are very important for hearing, because when damage is produced to the inner ear, these are the cells that first die. Now, what are they doing in hearing? These outer hair cells also have mechanotransduction channels in their apex, when this Meccano transduction channel, whose structure we still don't know, opened, the hair cell, this outer hair cell, depolarizes. And this depolarization produces movement of this hair cell. This hair cell shortens and elongates, and this produces mechanical amplification of all the movements within the cochlea. And this mechanical amplification is very important to produce amplification of the incoming sound, amplification of the signal, and fine-tuning of the basilar membrane, so that each part of our cochlea is sensitive to one specific frequency. And this mechanical movement is known as somatic electromotility. It only exists in mammals. It is a mammalian novelty, and it's based on a protein called prestin, which is here in green, highly expressed in the lateral wall of this hair cell. It is a motor protein which does not require neither ATP nor calcium to work, and it's highly packed on the side of the hair cell, and depolarization of that hair cell produces intramolecular rearrangements, and since it's highly packed, that intramolecular rearrangement is transduced into shortening and lengthening of the hair cell. And our work has demonstrated that this prestin protein was initially a transporter that accumulated mutations in mammals in the coding region of the protein to render now a protein that is highly capable of producing this somatic electromotility. I'm going to skip this. Now, so I basically told you very, very shortly how signals go from the ear to the brain. Now, the brain also sends information back to the inner ear, and it regulates or modulates the way we hear. And this top-down information goes all the way from the auditory system to the cochlea. And if we look at the cochlea, we see that the cochlea receives this red efferent innervation that makes synaptic contacts at the base of these outer hair cells. So at the place where somatic electromotility is produced. Now, for probably 50 years, it was known that this red medial-olivocochlear system is cholinergic. Acetylcholine is released at the base of these outer hair cells, and that the release of acetylcholine inhibits somatic electromotility. So it inhibits amplification. Now, in spite of the fact that it was known for 50 years that the main neurotransmitter was acetylcholine, it remained unknown the nature of the receptor present at the base of these outer hair cells. And both a ligand-gated ion channel and a G protein-coupled receptor was proposed, and there was a conundrum in the field until we went on and tried to clone that receptor. And the conundrum came from this. When one puts acetylcholine to the hair cells, you see these types of currents. First, you see a very small inward current that it's then surpassed by this huge outward current. So by cloning the receptor, we identified the molecular identity of that receptor and realized that it is a ligand-gated ion channel, which belongs to the nicotinic cholinergic receptor family. It is a pentameric receptor formed by two different subunits, alpha-9 nicotinic subunits and alpha-10 subunits. It belongs to the family of receptors that include serotonin-type receptors, GABA and glycine receptors. And with the cloning of those two subunits, we cloned the last nicotinic receptors that were missing in the entire family of nicotinic receptor subunits, which includes those present at the neuromuscular junction and those present in the central nervous system. And now we have the hair cell receptor. And over the years, we have now established that the acetylcholine, which is released at the base of the outer hair cells from this medial olivocochlear system, activates this alpha-9, alpha-10 nicotinic receptor. This receptor is highly calcium permeable. Calcium goes in, sodium also goes in, and this accounts for this small inward current. But this is coupled to nearby SK2 potassium channel that is activated by calcium. And when calcium binds to this receptor, it opens the receptor and potassium goes out from the cell. And that produces hyperpolarization and inhibition of that somatic electromotility. And this SK component accounts for this outward current. Now, of course, this synapse is not that simple. So over the years, we have completed, or not completed, but enlarged the knowledge we know. Here we have the alpha-9 and the SK component, and we have determined all the channels that are involved in the release of neurotransmitter, all the calcium channels, and presynaptic receptors like GABA-B receptors, and now also metabotropic glutamate receptors, which modulate, strictly modulate the functioning of this synapse. So I told you about the molecular structure of this receptor that mediates inhibition of somatic electromotility. Now, what is the function of this medial olivocochlear in hearing? Why do we need an efferent system that goes all the way from the brain to the ear and modulates the way we hear? Now, this is still an enigma, but over the years, we and others have tried to investigate these issues. Now, the hypothesis for medial olivocochlear function are several. It is a gain control system. It controls the gain of amplification. It is thought to mediate selective attention, to reduce auditory stimuli when we need to focus our attention, for example, in a visual task when we are reading. It inhibits auditory information. It controls masking from background noise so that we can filter all background noise and concentrate in what we are hearing. We know it shapes the normal development of the cochlea, and it also has been proposed to protect the inner ear from acoustic injury. So over the years, in order to tackle all these functions, we and others have generated several genetically modified mice, from knockout genes all the way to knock-in genes of the alpha-9 and alpha-10 receptors. And I'm just going to show you one result that we have obtained from this alpha-9 knocking mice. So we generated a mouse model in which we only mutated one amino acid in the transmembrane region of the receptor, losing for a threonine mutation just at the pore of the channel. And this produces a receptor that opens spontaneously, has a mean open time which is prolonged, so this receptor is opened longer. So this is a gain of function mutation, and we reasoned that if we introduce this receptor into a mouse model, we could generate a receptor which is opened more frequently, and therefore a medial olivocochlear system which was more enhanced, and analyze the consequences of an enhanced medial olivocochlear system to the hair cell. So we went on and contracted this animal. And this, what I'm just showing here, is patch clamp recordings from hair cells of these mice, from inner, I think these are inner, or it could be outer, both hair cells of this mouse. In black, you see the wild type mice, the outward current, which corresponds to the SK component. And in red, you see the knocking mice. You see that the currents are hugely expand. Yeah, this SK component has slower kinetics, and the current goes for a longer period of time. So, and then we did lots of auditory function physiology in the intact animal and really demonstrated that this animal has an enhanced medial olivocochlear system. And finally, what we went on and analyzed was if these animals were protected from damage produced by, is the last one, damage produced by produced by intense sound. So what we did was we traumatized the animals with sound, elevated sound, 100 decibels for 2 minutes. I need the last slide. Yeah, for 2 minutes, you can see, and we measure here threshold shifts. So we measured the higher amount of sound that we have to give to these animals in order for them to hear. In red and in black is the wild-type animals. You see there's like a 40 decibel threshold shift. You need to give 40 decibel more intense sounds for these animals to hear. But here in purple, the knocking animal has a reduced threshold shift. So these animals are indeed protected from noise-induced hearing loss. So this medial olivocochlear system, actually, one of the functions is to protect the animals from hearing loss. So I hope that I have given you some insights into the challenges of the field, preventing hearing loss, restoring hearing loss, and the genes and molecules that are involved in the entire process. And just the last one is to thank the people who did the work and thank the collaborators and the funding agencies, which are fundamental for all that I've presented. Thank you.